Hello and welcome to Science Research Weekly, Season 5, Episode 15. Expand your scientific scope. Get ready for algal microplastic remediation, cosmic large-scale structure, coffee and dementia risk, tales of outer membrane vesicles, and tumor microenvironment subtypes. Why a high R squared can be misleading, and finally, a MIRA award from the NIH. This will cover materials from May 8th to May 14th. Let's get started with our research articles. First up, remediation and upcycling of microplastics by algae with wastewater nutrient removal and bioproduction potential. Microplastics are everywhere and everywhere in the news, and so remediation is a hot topic. So in this paper, they look at algae, so specifically cyanobacteria, to actually remove microplastics up to over 90% within an hour with like 1 to 10 sort of ratio of microplastics per gram of biomass. They're actually able to even convert that cyanobacteria into plastic composites. So there is that. Next up, we'll zoom out to large-scale structure in Cosmos Web, tracing galaxy evolution in the cosmic web up to Z approximately 7 with the largest JWST survey. So that's the James Webb Space Telescope survey, and it looks at cosmic structure. Figure 2 is a good one. It's the count of the different sources, extra selection cuts to really have this wide scale mapping of the cosmos. Shifting gears here to JAMA, we have the paper Coffee and Tea Intake, Dementia Risk and Cognitive Function. So they looked at coffee and tea intake against dementia risk and cognitive function using the Nurses' Health Study and the Health Professionals Follow-Up Study. And they actually found was that as coffee use, so decaffeinated coffee didn't improve things, but caffeinated coffee use, they saw a drop, a greater in dementia and cognitive and better cognitive function. So the more coffee or even tea, caffeinated tea, was associated with lower risk of dementia. The real figure here is figure, was it at least figure 1A, ancient dementia hazard ratios. You see, starts at 1 for nothing, and it drops quite a bit. So it's like levels at about 3, and then more coffee doesn't help. Too bad. I'll probably have more anyways, but it looks about 3 cups of coffee a day, 2 to 3, really drops dementia risk by about 20%. So that's a really fascinating finding. And look, I don't need any more excuses to drink plenty of coffee in the morning, but I will certainly take it. That ends our normal articles. Let's switch to some preprints. First in archive, we have load identification in bistable spacecraft booms via parametric data-driven modeling. So there's these things called biostable tape string booms that help spacecraft self-deploy. So they employ storage strain energy, but it's hard to model that. So this paper looks at better modeling for that. So you can model it and then have more accurate deployment, I guess, or actually uses. And then there is the second article, like taking the path of least resistance, exploring the impact of LMM interaction on the creative process of programming. So, AI, there's a lot of studies onto, how creative their solutions are, but it's not really shown a lot. A bigger question is, how does generative AI affect the user's creativity? So they did some studies here, and what it looks like participants, using LLMs versus non-LLMs had shorter idea generation periods and fewer creative moments. And so while like the output, generally had more correct and functional code, their solutions had the same amount of ideas. So using LLMs did not make users more creative. Um, that's what it seems to take from that. So it's a good thing to keep in mind. This is a tool. It doesn't generate creativity on its own. That genuine creativity will ultimately come from humans, because that's really what LLMs are models made by humans trained on human data. So anyways, we can shift gears here to bioarchive, and we'll see our first of two papers. This first one is called Discovery and Characterization of OMVs produced by the bee gut microbiota. So OMVs are outer membrane vesicles, something I didn't know existed, but now I do, and I've expanded my horizons. These are little structures that bud off from bacterial cells, and they're essentially like they're transporters. They can transport different cargoes between cells and etc. And so not only was this paper looking at those, they were looking at those in the microbiota of the guts of bees. So even wider horizons. And then the second article was Tumor Archetype R, a modular framework to derive signature-based tumor subtypes. So it turns out that not only is there the tumor microenvironment that I've heard about a little bit, but you could actually use methods to detect subtypes of the microenvironment, and that might be important for tumor progression, etc. So that's what this package, R package, Tumor Archetype R, is for. It's unsupervised subtype discovery with generation of rank-based single sample classifiers. So there we go. All right, thinking about R, let's turn to our tools here. We have an R bloggers post from Learning Machines called Learning Data Science, Why a High R Squared Can Be Misleading. So typically a high R squared is an indication of a well-fit or accurate regression model, but that's not always the case. And so this short article goes into more about the meaning of it and how you can sort of be deceived if you're not thinking about it correctly. And new CRAN packages that might be of use, or at least of interest. We have Mars Runtime, Portable Mars Runtime Replay, QSR utils, R functions useful for community ecology. We have ROC serve comp, ROC-based methods for comparing survival distributions with right, left, and doubly censored data, and warm TH competence, warmth and competence detectors. With that, we'll close out our time here with one grant opportunity from the National Institutes of Health called Maximizing Investigators Research Award, or MIRA, for early stage investigators. And so it provides support for early stage investigators with a program of research that aligns with NIGMS mission. And so that might be of interest if you're an early stage investigator. With that, thanks for joining me for Science Research Weekly. As always, science on.